Okay guys, excuse me. What I'm going to do today is I'm going to explain to you how a motor nerve is able to cause skeletal muscle to contract. But before I do that, let's go over some basic anatomy. Muscles, their real function is to provide mobility, right? Move joints, and as protection. So what I'm going to do is I'm going to take you from the outside in, and then we get a little more specific. I'll show you another slide. Now, there's connective tissue that surrounds the belly of the muscle itself, so this whole muscle. That's called the epimysium or fascia. And the muscle actually contracts within the fascia. So the fascia doesn't contract. The muscle fibers itself contract within the fascia, and then the fascia thickens out and forms a tendon. So tendons connect muscle to bone. Ligaments connect bone to bone. So once you start moving deeper and deeper into the muscle, as you can see, these muscles are wrapped in fibers. So you got a bundle of muscle fibers that are wrapped together, and it's wrapped in what's called the paramecium. Then if you take a group of muscle fibers, as you can see, they're further wrapped, these individual muscle fibers, in a kind of a saran wrap called the endomysium. And then you have the muscle fiber itself. And the muscle fiber itself is basically made of just a few components. That muscle fiber, or sarcomere, right? It contains two contractile proteins, and we've talked about this before, called actin and myosin. So this is where the rubber meets the road here. These two contractile proteins are what ultimately produces muscle contraction. Now remember, the most important element in muscular contraction is calcium. Now we talked about the fact that the heart and blood vessels, arteries, they're made of muscle, and they get their calcium primarily from the blood. So embedded in the cells that make up the heart, for example, you have calcium leak and voltage gated channels embedded in the cell membrane. Now, in the cell membrane or sarcolemma of a skeletal muscle, there are no calcium channels in the cell membrane that makes up a muscle cell. The calcium is stored inside it. So calcium is stored inside that muscle cell, and it's stored in a place called the sarcoplasmic reticulum. So the sarcoplasmic reticulum stores the calcium. So there's no leak or voltage gated channels on the muscle cell membrane. So let's look at that just a little bit closer. So here is a muscle cell, a little bigger view, and then you have the cell membrane or sarcolemma of a skeletal muscle. Now, a couple of things you need to know. Number one, skeletal muscle cannot generate its own electrical activity like the heart can. Skeletal muscle relies on being stimulated to contract by a motor nerve, motor movement, right? So if you damage your spinal cord, remember that, excuse me, that it is conscious control of skeletal muscles. So the brain, the motor area of the brain, has to communicate with these motor nerves to stimulate a muscle to contract. And the spinal cord is the link between the brain and these motor nerves. So if you damage your spinal cord, any skeletal muscle below that injury, the brain's not going to be able to communicate with it, so you're not going to be able to stimulate those nerves. Therefore, you can't move those muscles. That's why people develop quadriplegia or paraplegia. So the skeletal muscle has to be stimulated by a motor nerve, right? But once stimulated, the membranes that make up the skeletal muscle can produce an electrical impulse. They can produce an action potential. But there has to be a stimulus for that muscle. And what stimulates the muscle? The motor nerve. Now, we talked about these actin and myosin filaments. This is really important. Remember, I just explained to you that calcium is stored inside a muscle. That calcium that's stored inside the muscle is stored in an area called the sarcoplasmic reticulum. So the sarcoplasmic reticulum kind of just surrounds all these muscle fibers, right? The actin and myosin filaments. And you can see there's like little offshoots to these guys. So the sarcoplasmic reticulum really does two things. Number one, it stores calcium. So embedded in the membrane of the sarcoplasmic reticulum are these little voltage gated calcium channels. Voltage implies electrical current. So when an electrical current travels over the sarcoplasmic reticulum, it opens up these voltage gated channels, and the calcium that used to be stored in there now floods into the actin and myosin filaments, and that's what you want. So the sarcoplasmic reticulum does two things. One, it stores the calcium used to contract skeletal muscle, and number two, it can actually transmit electrical impulses deep inside the muscle fibers, and that's what you want. You just don't want to contract the outer muscle fibers. Then what's the point of having muscle fibers on the interior of your muscle? You got to contract all of them. So the motor nerve that stimulates skeletal muscle to contract. So the motor nerve and the muscle fibers that it innervates, right, that motor nerve causes these particular muscle fibers to contract, is called a motor unit. So small motor units, MUs, they have one motor nerve supplies nervous innervation to a few skeletal muscle fibers, right? A large motor unit, then one nerve supplies many muscle fibers. So small motor units, fine coordinated movement. Large motor units, gross movement like walking, right? Fine motor units, small motor units, playing the piano. So let's get into now like kind of the interior of the muscle cell. So here you have a little diagram of the actin and myosin filaments. So you have actin protein, and then you have the myosin protein. And hanging off the myosin are these myosin heads. Now, in order to contract a muscle, and I'll show you in a video, to contract a muscle, you have to somehow connect this myosin head to this actin molecule. And sitting on that myosin head is a molecule of our buddy ATP. So you somehow have to connect the myosin head to the actin molecule. And once you've connected the myosin head to the actin molecule, you will take ATP that's sitting on that myosin head right there and break it down to ADP plus a phosphate plus energy. And that energy that's released when you break off that third phosphate cause that myosin head to swivel. And because it's connected to the actin molecule, you pull the actin molecule along with it. And when you're pulling actin, you're contracting. Now, unlike the heart, the stronger the stimulus, the more muscle fibers are activated. And the more muscle fibers that are activated, the more connections you can make to actin and myosin. So if you make more connections between actin and myosin, that increases the force of contraction. And just like the heart, if you add more blood to it, you stretch the actin and myosin, and it lines up better, so you can make more connections between actin and myosin. So skeletal muscle works the same way. If you stretch a skeletal muscle, you line up the actin and myosin better. The more connections you make, the more powerful the contraction. And that's Starling's law. So what I'm going to do now for you is, I showed you the video on how a cell, or how a motor nerve, creates an electrical impulse, right, an action potential. So let me diagram this for you. The end of a motor nerve, right, the presynaptic membrane stores the neurotransmitter acetylcholine. So acetylcholine is the neurotransmitter for skeletal muscle contraction, always. So once a motor nerve is stimulated, it's going to cause that electrical impulse excuse me, to travel down the axon of that motor nerve. And when that electrical impulse travels over the presynaptic membrane, it's going to cause the release of the neurotransmitter acetylcholine. Now, let's give you an idea of where you're at. Remember, nerves and other nerves don't touch. Nerves and muscle cells don't touch. They form a synapse. So this is the synapse for skeletal muscle contraction. It's also called the myoneurial junction. Right? So you know some of these terms. So how does a motor nerve communicate with skeletal muscle? That nerve is stimulated, produces an electrical impulse with sodium, potassium entering and leaving. That electrical impulse travels over the end of that axon, that presynaptic membrane, and causes the release of acetylcholine. Now, embedded in the muscle cell membrane at the myoneurial junction, you're not going to believe this, are these little receptors called acetylcholine receptors. And you'll never believe what binds to acetylcholine receptors. You know, you guessed it. Acetylcholine. So again, skeletal muscles cannot generate their own electrical activity. But once that skeletal muscle is stimulated by the neurotransmitter acetylcholine, it can propagate an electrical impulse. So it can spread that electrical impulse over the muscle cell membrane. So embedded in the muscle cell membrane are sodium and potassium voltage gated channels. So what this does is allow that electrical impulse to travel over the muscle cell membrane, and that's what you want. And what caused it was the release of that acetylcholine. And then acetylcholine binds to these acetylcholine receptors. And these acetylcholine receptors are nothing more than sodium leak channels. So as a result of acetylcholine being released, when acetylcholine binds to those acetylcholine receptors, it's going to cause the opening and closing of sodium and potassium voltage gated channels. So it's going to cause sodium to leak in, and it's going to reach a critical voltage inside that muscle cell and then activate the sodium potassium voltage gated channels along that muscle cell membrane. So observe.
Traction coupling consistent.
So acetylcholine is going to be released. It's going to bind to those little acetylcholine receptors. Those are sodium channels. Sodium leaks in, and then it will activate sodium and potassium voltage gated channels along the muscle cell membrane. Now, what allows the electrical impulse to travel deep inside the muscle is the sarcoplasmic reticulum. So that muscle cell membrane is going to develop that action potential, and then it's going to travel over the sarcoplasmic reticulum. What's stored in the sarcoplasmic reticulum? Calcium. So when that electrical impulse that was caused by the release of the neurotransmitter acetylcholine binds to those acetylcholine receptors, that electrical impulse travels over the muscle cell membrane, and then it's going to open up voltage gated calcium channels in the sarcoplasmic reticulum. And things, as we know, always go from high to low. So calcium that was stored in the sarcoplasmic reticulum, once the electrical activity travels over the sarcoplasmic reticulum, it's going to open up voltage gated channels on the sarcoplasmic reticulum, and calcium is going to flood into the actin and myosin filaments. Now, so let's look. We got the myosin head, and sitting on the myosin head is a molecule of ATP. Somehow, you've got to connect that myosin head to that actin molecule. The problem is, in a muscle that's not contracting, this thing that looks like a purplish blue rope, this is the protein tropomyosin. And in a uncontracted muscle that's not contracting, tropomyosin covers the binding sites on actin for myosin. So if tropomyosin is in the way, the myosin head can't bind to the actin. This is where calcium comes in. As you can see, these things that look like partially chewed banana bubblicious, this is what's called the troponin complex. And it sits on the tropomyosin. So when calcium floods into the actin and myosin filaments, because of the electrical impulse and the opening of those voltage-gated calcium channels on the sarcoplasmic reticulum, calcium is going to flood in, and calcium binds to troponin C on the troponin complex. And when calcium does that, it causes the tropomyosin molecule that was covering the binding sites for the myosin head on the actin molecule. The actin molecule looks like partially chewed beer nuts here. When that happens, it causes the tropomyosin molecule to twist out of the way, and now those binding sites on the partially chewed beer nuts right here, they are open, and the myosin head can bind there. So you got calcium binding to troponin C, and it's going to cause that tropomyosin molecule to twist out of the way. And when it twists out of the way, it's going to open up those binding sites on the actin molecule for the myosin head. And there's a natural attraction between the little blue chicken leg and the partially chewed beer nut, the myosin head to the actin molecule. So observe. So now you've got the myosin head bound to the actin molecule. What's sitting on the myosin head? A molecule of ATP. So when the myosin head binds to the actin, it's going to cause that third phosphate to be hacked off. And when that third phosphate is hacked off, it releases heat and energy. And that energy that's released, will cause the myosin head to swivel, and now it's connected to the actin molecule, so it's going to pull the actin molecule along with it. And again, when you're pulling actin, you're contracting. So it's going to cause that myosin head to swivel a little bit, and it's going to pull the actin molecule along with it. Now, if you want to relax the muscle, what do you got to do? Well, you got to stop the motor nerve from releasing acetylcholine. So you say to yourself, okay, I want to relax my muscle. So you stop the nerve impulse, right, the electrical impulse in the motor nerve. Right? So let's go back. Once you stop the electrical impulse, there is a enzyme floating in the gap between the motor nerve and the muscle cell membrane called acetylcholine esterase. And that enzyme degrades the acetylcholine. So if acetylcholine isn't there anymore, sodium won't leak in. And if sodium doesn't leak in, then you're not going to be able to propagate an electrical impulse over the muscle cell membrane. So the electrical impulse over that muscle cell membrane stops. Now, you probably heard of a condition called myasthenia gravis. Myasthenia gravis is where it's an autoimmune disease where the immune system starts attacking and destroying acetylcholine receptors in skeletal muscles. So myasthenia gravis literally means grave muscle weakness. So there's drugs out there that will inhibit acetylcholinesterase. Now watch. If you have less acetylcholine receptors, that means less acetylcholine can bind. So you don't want to be degrading that acetylcholine, right? Otherwise your muscles become weak. So that's why they give them a drug that inhibits that enzyme. So that's used how they treat myasthenia gravis. Now, you stop the electrical impulse by removing the acetylcholine from the acetylcholine receptors, right? So the electrical impulse is stopped. But you still have myosin connected to actin. So you got to break that connection. So how do you break that connection? Well, remember that when you take ATP and you break off that third phosphate, that's going to leave ADP plus little phosphate. So As ADP starts building up in that muscle cell, that's going to stimulate the enzymes of metabolism to break down more glucose or fat, the fuel that's most readily available, remember? And metabolism's going to speed up so that you can pop on that third ADP or third phosphate to ADP and recycle ATP. So when you make a new ATP molecule and it rebinds to that myosin head, it will cause the connection between actin and myosin to break. Also, you got to remove that calcium. So calcium that was bound to troponin C on the tropomyosin molecule, that has got to get pumped back into the sarcoplasmic reticulum. So when you do that, when the muscle relaxes, that activates the calcium pump in the sarcoplasmic reticulum, and that pumps the calcium back into the sarcoplasmic reticulum, and your muscle has relaxed. So that's kind of actin and myosin and muscle contraction. So where you need to start this explanation is you need to explain to me in the video that I posted on how a nerve generates an electrical impulse, right? That electrical impulse then causes the release of acetylcholine, and then you can explain to me how calcium causes the muscle to contract. So I hope that helped.